Hey Paul, I have a doctor's appointment today, so I'm gonna have to log off a bit early, but I'll watch the rest of the lecture on YouTube later if that's okay.
Okay, yep, that's fine.
Thank you.
Oh, that's why. Okay. All right. Ugh. Mm. Okay. How are you guys doing today?
Good.
How are you guys holding up in terms of pandemic land?
It's getting annoying.
Just now?
Well,
Just
no,
now
but
getting annoying? How long has it been annoying?
a long time.
So interesting thing happened in the last 2 weeks. We, as a nation, were treated to our first 2 mass shootings in over a year. Who knew that one of the
I did.
consequences, what's that?
Oh, sorry. I thought you were asking who knew that. Go ahead.
Oh, you knew that. Yeah, I didn't realize that it has been that long since we had a mass shooting. And I guess, people are talking about the COVID pandemic, and it's like one of the upsides is a decline in other communicable diseases. So instances of flu have gone down. A guy that I bicycle with, he has a second job during Christmas time. He is a Santa. He hires himself out as a Santa and does kids parties and events at stores and things like this. And he told me a couple of months ago that this is the first Santa season in his entire career as a Santa when he didn't get sick. And he is rabidly anti-mask, but he was required to mask during all of his public appearances as Santa. And he attributes the fact that he didn't get sick from other things due to masking, surprisingly enough. So 2 of the weird kind of positive outcomes of going through a pandemic is a severe decline in mass shootings, and Santas don't get sick as often as they used to. And obviously rates of flu have also gone down. Flu transmission, regular standard flu. Pandemic updates. Sounds like Missouri is opening up, I guess, tier 2 and tier 3, which I think tier 3 is just everybody. We're available to get vaccinated starting next week, from what I have heard. If you haven't signed up with Clay County Health or Operation Safe, then by all means do that. Get vaccinated. The more of the campus is vaccinated, the sooner we can kind of get back to normal. I realize all of y'all are seniors, right? Every one of you are seniors.
Yeah.
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 Well, okay. The fact that we're probably going to have in-person classes in the fall probably is just nothing but rubbing salt in the wound to you. But we do look forward to a day when we can get back to business as usual. I have signed up at Clay County Health and Operation Safe, both in an effort to get vaccinated as soon as I can. So we'll see how that goes. We'll see if next week is actually going to pan out to be what it is. Clay County Health has already, they sent me a thing. I think, did I tell you about this? They sent me an email 2 weeks ago, basically saying, you're eligible to get the vaccine, sign up here. And then you click on the link and it takes you to the same questionnaire that you answered when you signed up to be on the waiting list to begin with. And it asked you all the same questions, which basically indicated that I was in tier 2, when they were still only doing tier 1B. And I was like, why did you send me a link saying that I was eligible when you knew darn well that I was not eligible? And I got to the end of it and said, you're not eligible. I'm like, I know I'm not eligible. I've been following the news. Come on, people. I've now filled out both the Clay County thing twice and the Operation Safe thing twice, because people keep sending me things saying that I need to fill them out again. So we'll see what happens ultimately. I'm quite looking forward to a shot in the arm. Right, questions about energy and gas exchange, the stuff that we've been covering up to this point. All right, then I shall share my screen. We finished up last time talking about the effect of carrying offspring or eggs and its effect on ventilation when you have a big reproductive load taking up space in your abdomen. Obviously, that negatively affects your ability to use your lungs. And these are images showing you how lung volume has decreased in the presence of this developing embryo in one of the shingleback skinks from, I believe, Australia. So there are all kinds of constraints, and one of the things that we'll talk about a lot, both during this chapter and in all the future chapters, is kind of the nature of constraint, the idea that you can't be perfectly adapted in all ways to all particular ecological or physiological challenges. And so an organism, as it appears, is actually a balance of a lot of competing selective pressures acting on individuals over long periods of time. And so we would not expect to see perfectly adapted animals to their environment, because adaptation among lots of competing challenges ends up resulting in compromised organisms, not perfectly adapted organisms. So this is just one example of that. In organisms, in reptiles and amphibians that are long, have long bodies, oftentimes this results in lung reduction. And in snakes and amphisbaenids, it's a lung reduction to the point that only one of them is still present and functional in a sense. In snakes, the right lung is the main lung and the left lung is reduced, or in some cases completely lost. And in amphisbaenids, the right lung is completely lost, so they rely on the left lung. So it's just evolutionary happenstance as to which one got lost, but the one that remains is the one they use for gas exchange. And the interesting thing in snakes is that the lung is really long. It goes down the entire length of the body, and it's divided into two parts. One is called the saccular lung, and one is called the vascular lung. The saccular lung is basically just a kind of a dead airspace, and most of the gas exchange occurs actually in the vascular lung. So if you look at inhalation and exhalation and its effect on oxygen concentration, when the snake exhales, it's getting rid of a lot of air, so oxygen concentration goes down. When it inhales, it goes back up. As the snake then essentially is in the time period between inhalation and exhalation, the total oxygen content of the lung goes down gradually, because oxygen is diffusing out of the lung into the vascular spaces around the lung, and depletes oxygen level until it goes through and inhales again. And if you've ever watched a snake breathe, you will see that it spends a lot of its time just sitting there, presumably not moving. And then all of a sudden you'll see its body contract and get smaller. That is the animal actively exhaling. And then you will see its body puff up again, inhaling, and then it will go back to rest. And so that's what you're seeing here during this relaxation phase, is that period between breaths. And that period between breaths can be quite long. And then it becomes noticeable that the animal is actually inhaling and exhaling. And then the same thing happens with the opposite gas that is of importance here. As oxygen concentration is going down, CO2 concentration is going up. And you see that these levels fluctuate a lot in the vascular lung, but they don't fluctuate very much in the saccular lung, which indicates that the saccular lung really isn't involved in gas exchange per se. Turtles are a special problem in terms of ventilation, and crocodilians are kind of in the same case. They're much less flexible in their torso than other organisms are. In crocodilians, the ribs are very static. They actually appear as these kind of plates that go out to the side, and they don't go all the way around the rib cage the way ours do. And as a result, it's difficult to ventilate the lungs very much. And so in both of these cases, both with crocodilians and turtles, they ventilate their lungs basically by moving around their viscera. And so in the case of crocodilians, they move their liver around to create the negative pressure that allows inhalation. And in the case of turtles, the viscera are moved by leg movements. And so the animal will actually move its legs into the space underneath the carapace and out of the space underneath the carapace. When the animal moves its leg inward, that presses its viscera up on the lungs, forces the lungs to deflate and exhale. And then when it extends its leg out again, it pulls the viscera back, creating that negative pressure and allowing the turtle to inhale. You will also notice on turtles that they also do some gular pumping, where they're using this gular pouch under their neck to help ventilate their lungs as well. So that's kind of whole organism ventilation and some of the complications of whole organism ventilation. Do you guys have any questions about that? Okay. So eggs, we've already talked a little bit about the problem with eggs. Basically, this problem of gas exchange going through either fluid barriers, so for example, diffusion through the albumin, the protein part of the egg white, if you want to think about that, versus reptiles that wrap their eggs in a strong structural shell. And as a result, that shell has these little tiny channels that allow oxygen to flow through the shell. If you ever look at a shell under a microscope, you'll see that it has tons of little pores on it. I actually have an ostrich egg in my office. Why, I don't know. It's big. And if you ever come by my office, you can see at one end, they have drilled a hole that allowed them to get rid of the contents of the egg so that it doesn't smell terrible. But when you look at it from the side, and you can't really see this, but like I said, you can see it if you come by my office, this eggshell is actually quite thick and heavy. And it poses a significant barrier to the movement of materials into and out of the egg, but it has the same level of porosity that you see in reptile eggs as well. Pores, tiny microscopic pores that allow airflow to reach the inner membranes of the egg. So once again, we've talked about earlier in the lecture last time, we talked about the challenge of breathing in eggs, and we also talked about how some amphibians have specialized structures that allow them to get gas exchange inside of eggs. So this is just a reiteration of that challenge. So when we think about the circulatory system of organisms, the heart structure of amphibians and reptiles is different in many cases from our heart structure, in that the chambers of the heart are not completely divided. And as a result of this, you get quite a lot of blood mixing. And so amphibians have the ability to both get gas exchange through the lungs as well as through the skin. And so as blood leaves the heart and goes to both the skin and lungs, that's where you get oxygen exchange. But the relative amounts of oxygen uptake and CO2 release change depending upon whether you're talking about an amphibian circulatory system or a reptilian circulatory system. So you can see that most of the oxygen uptake and CO2 release in amphibians is done cutaneously. And one of the problems with this is that there is a lot of blood mixing in the hearts of amphibians because the hearts of amphibians don't have complete division between the atria and the ventricles. And so when we look at an amphibian heart, you can see that they have a right atrium and a left atrium. The right atrium is receiving blood from the body, so this is going to be oxygen poor blood. The left atrium is receiving blood from the lungs and skin, and so that's the oxygen rich blood. And both of these atria dump into a common ventricle. And so the problem with dumping blood into a common ventricle is that it's in the ventricle where you get the mixing of oxygen rich blood with oxygen poor blood. It would be much more efficient if you could just take this blood that is oxygen poor, pump it out directly to the pulmonocutaneous artery, the artery that goes to the lungs and the skin, take that deoxygenated blood, pump it out to the lungs and skin where it gets oxygenated, and take the oxygenated blood and pump it out to the rest of the body without any sort of mixing. But the design of the heart, the way this heart has evolved in amphibians, you have the possibility of mixing. One of the ways in which they have gotten around this common ventricular chamber is through the development of what we call a spiral valve. And if you ever dissect a shark, the stomach of the shark has a spiral valve. What that does is it basically, it's a series of membranes inside of the artery that essentially take a single tube and divide it up into a segmented tube that's segmented longitudinally. And then it spirals around, so it actually does look like a spiral. So these membranes twist. And between these trabeculae, so inside of the heart, you will always see these ridges. If you've taken anatomy and you've actually dissected a heart, you'll see that there are all these internal ridges of muscle inside a human heart and also inside the heart of amphibians. The combination of these trabeculae, these ridges, coupled with the spiral valve, actually works to reduce the amount of mixing. And so what you will see is that the oxygen-rich blood coming from the atrium interacts with these trabeculae, and these trabeculae direct the oxygen-rich blood to a part of the spiral valve that will then ultimately direct oxygen-rich blood out to the body. And in the same way, the trabeculae in the right-hand side of the ventricle will direct it towards the right-hand part of the spiral valve, and that will send more of the deoxygenated blood out to the pulmonocutaneous artery, out to the skin and lungs to become oxygenated. So while there is mixing of oxygenated and deoxygenated blood, combination of the structure of the heart and this spiral valve in the aorta serves to reduce that mixing to a certain degree. So that's one of the challenges of the amphibian heart, because the segmentation of the ventricle is not present at all. You have the possibility of blood mixing, and the spiral valve would presumably be an adaptation to reduce that. What happens in the reptile heart is you don't have complete separation in the ventricle, but you have partial separation in the ventricle, such that you have this massive muscle that basically separates the right ventricle from the left ventricle. Remember that these are all all views of the heart looking at the animal from the ventral side. And so everything is flipped. The right side of the animal's body is on the left side of the image. So the right side is the pulmonary side, the left side is the right side receives blood from the body and sends blood to the lung, and the left side receives blood from the lung and sends blood back out to the rest of the body. And so the ventricle is subdivided, but it's not a complete subdivision. So as a result, we have different sections. The cavum venosum is this section up here at the top above this partial partition. The cavum pulmonale is the section of the ventricle that receives blood from the body. And then the cavum arteriosum is the portion that receives blood from the lungs. And what happens during this is during atrial contraction, blood moves from the atria down into the ventricles. And when this happens, the valves that are at the juncture between the right atrium and the left atrium and the common ventricular space, these valves basically, essentially with the flow of blood, they close up the opening between the two ventricles. So the action of pushing blood out of the atria essentially closes the two halves of the ventricle off from one another. This then prevents the mixing of oxygenated and deoxygenated blood. Then what, pardon me. Then what happens during ventricular contraction is there is a valve between this portion of the ventricle here. The contraction here essentially closes off that valve, closes off the valves to the atria, and essentially through a valve that separates these two portions of the ventricle, it once again keeps oxygenated blood in one half of the heart going out to the body to provide oxygen to the organs, and it sends the other half of the blood off to the lungs to become oxygenated. And so through a combination of valves and muscular contraction, the two halves of this incompletely divided heart essentially keep mixing of deoxygenated and oxygenated blood to a minimum. So this is generally what happens in the hearts of most reptiles: partial division of the ventricle versus no division of the ventricle in amphibians. How are crocodilian hearts different from these two styles of heart? And I forgot to do something the other day because I got busy with other things. Give me a minute. So work on answering that question. How are crocodilian hearts different from these other hearts?
Isn't the crocodilian heart more similar to like that of mammals with the complete?
It is. And what is that difference?
The like complete ventricular separation, like the wall.
Yep, complete ventricular separation. Yeah, so I couldn't remember if I had posted the energetics and performance slides, but they are up. So I went and checked that. Faulty memory, I guess. So one of the advances that has occurred in the hearts of crocodilians is you now have this complete separation where the right hand side of the heart is completely separated from the left hand side of the heart with this muscular septum that keeps them separated. And the only place where you get the possibility of blood mixing is that oftentimes there are arterial shunts. So remember that the difference between, what is the difference between an artery and a vein? This is something that students, when they first walk into an anatomy class, make a mistake in defining what an artery is and how it's different from a vein.
Arteries carry blood away from the heart and veins carry blood towards the heart.
Very good. What's the common mistake that beginning students make?
Arteries carry oxygenated blood, but the pulmonary arteries and veins are the problem with that definition in
Yes,
humans.
very good. You've taken anatomy, haven't you?
Yes.
That is exactly the kind of information that I would assume an anatomy professor would impart to their students. So whether you're an artery or a vein is determined by whether you're carrying blood away from the heart or whether you're carrying blood back to the heart, regardless of whether that blood is oxygenated or not. And so you can see here that there is a left aorta and a right aorta. These are arteries that are carrying blood away from the heart. And you will see that there is an arterial shunt between the left and the right aorta. The right aorta is carrying blood. Wait, why is that labeled that way? One of these aortas is carrying blood to the lungs, which is deoxygenated blood. One is carrying blood to the body, which is oxygenated blood, but there's usually an arterial shunt between these. And as a matter of fact, when you're a baby, you actually have a shunt that goes between your two atria, because when you're a fetus, there's no need to send a lot of blood supply to your lungs because your lungs are not functioning. And so there's a shunt between your two atria that allows blood to be basically circulated more to the rest of your body than to your lungs, because your lungs aren't being used as a respiratory organ. And when you're born, that shunt shuts up. And actually, if you have a mammalian heart and you put your pinky fingers into the two atria, you can feel around at the septum that separates the two atria, and there's a little thin spot. So most of that wall is relatively thick, and then there's a little thin spot. So these shunts between different sections of the heart, or between the aorta coming out of the heart, are not uncommon. And so the crocodilian heart is structured much in the way bird and mammalian hearts are structured. And so normally you have the right atrium receiving blood from the body, coming into the right ventricle. That blood is then pumped out to the lungs. The lungs oxygenate the blood. That blood coming back from the lungs enters the left atrium, comes into the left ventricle, and then is pumped out through the aorta here. And so what you will oftentimes see is shunting between the pulmonary artery and the aorta. So this idea of cardiac shunts becomes important because it allows an animal to control what parts of the body it's sending blood flow to. And this is oftentimes governed by how much resistance there is in the circulatory system. So when we were talking about thermoregulation, we talked about the fact that animals can relax the muscles in their arterioles. And so the arterioles are the second to the smallest division of the blood vessels before you get to capillaries. And what makes a capillary different from an arteriole is capillaries are not wrapped in muscular layers, and arterioles are the smallest vessels that are wrapped in smooth muscle. And so you can control the blood flow to a particular part of your body by either relaxing or constricting your blood flow with these circular muscle layers that go around your arterioles. If you have contracted those circular smooth muscle layers, it restricts blood flow to the capillaries, and so you reduce blood flow. But as you reduce blood flow, you're also creating a resistance to the blood flow. And so you're basically kind of like putting a stopper in that arteriole, and then the blood backs up and it provides resistance in terms of resistance to blood flow in that particular area of the body. That then backs up to the system and can back up all the way to the heart. So if you have, let's say that you're cold and you're trying to conserve core temperature, you can constrict the arterioles that go to the capillaries of your skin. That will prevent blood flow through to the skin. As a result, you're not radiating a bunch of heat out to the environment so that you're conserving more of that heat internally. But what that does is it causes resistance in those arteries going to the skin capillaries, and that pressure backs all the way up into the aorta here, such that when the left atrium dumps blood into the left ventricle and the left ventricle contracts to send blood out through the aorta, there's more resistance to sending blood that direction because you've constricted your arterioles in that part of your body. In the same way, you can constrict the arterioles in your lungs. That would then create resistance in the pulmonary artery, which then creates resistance when you go to contract your right ventricle to pump blood that direction. So if you have a shunt that goes between these two things, you can actually move blood from one part of your body to another part of your body here at the heart through basically creating resistance in other parts of the circulatory system. So if you decrease systemic resistance, systemic resistance is resistance caused by your body increasing contraction of these small muscles in your arterioles, then it basically sends blood from the right hand side of your, decrease resistance on this side allows blood from the right hand side of the heart to move to the left hand side of the heart. Decrease pulmonary resistance allows the opposite move of blood. So if you have right to left shunting, basically it's because you've decreased resistance on this side such that blood from the deoxygenated side of the heart can go and enter the oxygenated blood. And if you have decreased resistance in the pulmonary side, then blood will flow the opposite direction. So this cardiac shunt occurs here. And so you have the pulmonary arteries, the pulmonary artery going to the right lung, and the left aorta is going to the left lung, and you have a shunt going between these two aorta. If you need more blood flow to flow into the lungs, you can reduce resistance on this side, and you'll see this movement of blood from the left side of the heart to the right side of the heart through the foramen of Penisi, which, if you looked at this FP here, that's what that's referring to. And so this is basically just where the shunt is located and how it's operating. Increased resistance, sorry. Yeah, increasing resistance sends more blood out to the system. Increasing resistance in the pulmonary side sends more blood flow out to the systemic part of the body. Lower resistance sends more blood flow to the lungs. And so once again, just allows the animal to control where blood is moving. Right to left shunting speeds warming by increasing systemic blood flow. Both things can stabilize the ratio of oxygenated to deoxygenated blood, and left to right increases blood flow to the heart, because one of the things that happens when blood is flowing out here, coming off of the right aorta, are a bunch of, is a single actually blood vessel that divides immediately after it comes out of the right aorta and supplies blood flow to the heart. And so you can increase heart rate and heart function by diverting blood to the heart itself. So if you are in the process of trying to raise your body temperature, what you want to do is you want to get as much blood flow as possible out to the skin if you are one of these organisms that regulates its body temperature through behavioral means of thermoregulation, things like basking. And so if you basically shift blood flow so that there's more blood flow going out to the skin, you basically will warm up more blood. That blood then becomes circulated back into the heart and then pumped out to the body. And so this is something that you would do as part of behavioral thermoregulation, is shunting blood towards systemic circulation. And when you see a lizard basking or a snake basking, they're not actually engaging in a lot of activity. They're basically laying still someplace and essentially reducing the blood flow needs of other parts of the body so that they can send blood out primarily to the extremities and the skin. So shunting in animals. This combined with countercurrent mechanisms, which we talked about back in thermoregulation, actually helps organisms raise their body temperature. So metabolism is another way in which an organism can raise its body temperature. And there are different forms of metabolism. So if you've taken a physiology class, you've talked about some of these. So essentially, your metabolism, where you get your energy to both raise body temperature and also, run away from a predator, is through ATP. And so there's a lot of energy stored in these triple bonds that bind phosphate molecules to ADP. And when you strip those phosphate molecules off, it releases that energy, and you basically take ATP, remove a phosphate, converting it into ADP. And if you want to store energy again, you have to absorb some energy to reattach that phosphate. So ATP is found a lot in skeletal muscles. That's where, for amphibians and reptiles, that's where most of their metabolism is going to be centered. And phosphocreatine is one of the compounds that also stores phosphate. And so as a result of this phosphocreatine in skeletal muscle, you have a lot of ATP being stored. And so phosphocreatine is used to generate ATP and store it in the skeletal muscle. And this storage of ATP in amphibians allows you to accomplish about 100 muscle contractions. And so when you are converting phosphocreatine to ATP, that's a way of getting a hit of quick energy out of your phosphate storage mechanisms. But once that is exhausted, your choices are then to go into normal kind of oxidative, what we call aerobic metabolism, or go into glycolytic. And glycolytic is spelled incorrectly on this slide. And so one of the things that you'll see if you ever go to a frog jumping contest, have any of you ever been to a frog jumping contest? Elizabeth Ellis is smiling as if she has not been to a frog jumping contest, but maybe wants to go to a frog jumping contest. Frog jumping contests are where you take frogs, go out and catch a bunch of frogs, and then you put them in an arena, and you see basically who can jump the furthest in a particular time period. And one of the challenges of being a frog jumping is that you run out of ATP relatively quickly because you're largely relying on the stored ATP in skeletal muscle to get this burst of energy. And you will see for most amphibians and reptiles, they rely on short bursts of energy to escape predation, and they are not particularly good at long sustained efforts. And it's because after they've exhausted this stored ATP in their muscles, they then start having to break down either fat or glucose to create more ATP. And that's a slower method of getting energy available to your muscles than just by taking ATP that has been stored in skeletal muscles in the form of phosphocreatine. So this is the, wow, lots of typos in this presentation. So oxidative is more efficient, but is slower to respond. If you take a mole of glycogen, you get 35 moles of ATP. Glycolytic is quicker to respond, but it's much less efficient. It basically gains you 3 moles of ATP for the 1 mole of glycogen that you get out of it. So once again, you have 2 methods of ATP production here. One that is more efficient but slower to respond, one of which responds very quickly but generates less energy total. And also when you look at the byproducts of this, these 2 modes of production, the byproducts here are relatively benign, carbon dioxide and water, whereas the byproduct of glycolytic ATP production is the buildup of lactic acid. And so how many of you are athletes? I can only see 2 of you, and Kiara looks like she's holding somebody for ransom in some bunker somewhere. So Kiara's an athlete. Yep. Quinn, you are also an athlete?
Yeah.
And
Me
Kelsey?
too.
Yeah, so Kiara plays basketball. What do you do, Quinn?
Volleyball.
Volleyball. And Kelsey?
Swimming.
Swimming. Okay, so I've never played any of those sports, actually. I'm a terrible swimmer. When you have a really hard workout that is really highly aerobic, one of the dangers that you have to guard against as an athlete is essentially getting to the point where you leave aerobic respiration and you start entering into glycolytic respiration. Do you guys have this problem in any of your sports? Volleyball, probably not. Basketball, probably not. Kelsey?
The whole point of swimming is to not breathe, so yeah.
Oh, so you do have this problem. Okay. So yeah, I didn't think about it from that perspective, which is maybe why I don't like swimming. If you're an endurance athlete, one of the problems that you have is keeping glucose available enough that you're using glucose primarily for your energy supply rather than using stored glycogen. And so if you're running a marathon or doing a long endurance race on a bicycle or something like this, one of the challenges that you have is leaving kind of oxidative ATP production and going into glycolytic ATP production, because then what happens is you start building up lactic acid in your muscles. And that ultimately is a less efficient way of utilizing energy from the standpoint of moles of ATP, but the lactic acid can also start to do damage to your body in terms of, in this case, degrading muscle, because you now have this buildup of lactic acid. And so generally you want to stay in oxidative ATP production and avoid glycolytic. The problem with herps is that oftentimes when they need energy right then. And so reptiles in particular, and amphibians to a certain degree as well, depend upon glycolytic production of ATP a lot more than other organisms, because they need a quick dose of ATP to get them through whatever short-lived intense activity that they're going to be involved in, which oftentimes is short sprints to try and avoid a predator. As a result, they are not particularly efficient at using energy, and this energy gets used up pretty quickly. And now I'm unable to move. So when you look at muscle, you will see, for example, red meat and white meat in a chicken. Have any of you, how many of you have had duck or goose as a meal? Anybody? Okay, so if you ever eat duck, one of the things that you will notice about the breast meat of a duck is that it is much juicier and tender than the breast meat of a chicken or a turkey. And the reason for this is that ducks are out there in nature and they're using these muscles for flight. As a result, those muscles are being used actively for the movement of the organism. And as a result, they are rich in myoglobin. And so the difference between red muscle and white muscle has everything to do with the type of muscle fibers that are there and the types of compounds that are there to, for example, assist in moving oxygen around. And so I don't really like breast meat of chickens and turkeys because I find it amazingly dry in most cases because it's not cooked properly. And even when it is cooked properly, it's basically made up of a different set of fibers and molecules that are found in that muscle, which basically make it tougher than drier than dark meat, which is on the legs and thighs. Turkeys and chickens use their legs and thighs all the time because they're basically running around and they don't ever fly. Whereas ducks both use their thigh and leg muscles, but they also use their breast muscles. As a result, they have more dark meat, if you want to think of it this way, in their breasts than turkeys and chickens do. And the reason that it's dark is that it is rich in myoglobin, which is this oxygen carrying pigment that occurs in muscle. And these are oxidative fibers because this is what you find in muscle that is used a lot. White muscle, on the other hand, are muscle fibers that are going to use glycolytic ATP production a lot. And one of the things that you'll notice if you ever eat a reptile, how many of you have eaten a reptile? Had turtle soup, alligator tail, snake. Oh, Kiara, you've had alligator tail?
Alligator.
Oh, where'd you have alligator?
Down in Florida.
Yeah. What was that dining experience like?
It's kind of.
How did that meat compare to other meats you've had?
I actually really like alligator.
Yeah, but what do people say about alligator oftentimes?
Tastes like chicken.
They say it tastes like chicken. Once again, because when people associate chicken, they're oftentimes talking about breast meat. And so you're basically talking about this white flesh, which is made up of these glycolytic fibers, which are not being used on a regular basis. As a result, they're not rich in myoglobin. And so when you look at the flesh from something like a turtle or an alligator or something like this, it much more resembles the white meat that you're familiar with from birds. And so the type of muscle that you have is going to be influenced by how much that muscle is used in sustained kind of aerobic activity. Use your muscles a lot, they build up rich supplies of myoglobin and a different type of muscle fiber that is going to use a different type of ATP production. And if you don't use your muscles in this aerobic sort of way as often, you're going to have muscle dominated by a different type of muscle fiber, and that is going to be less rich in myoglobin and have different characteristics as a result of it. Amphibians and reptiles tend towards the glycolytic types of muscle fibers, with a couple of exceptions. The vocalization muscles in amphibians are generally going to be oxidative. They are associated with the lungs and with the upper respiratory system. And one of the things that you find with vocalization in amphibians is it is a very energy intensive sort of activity. And so you would expect to have red muscle, this oxidative type of muscle, in those areas that are using a lot of energy over long sustained periods of time. I think I keep forgetting to tell you, but frogs have begun calling. They started about, I think it was March 11th. Yeah, March 11th. I was out on a bike ride on a Thursday evening, and I heard the first calls of the year on the 11th. And if you go out to a frog chorus, which we will do in the coming weeks, what you will hear is you will hear frogs that are essentially calling nonstop. They're calling during the daytime, they're calling at night, and this is the time when males are advertising to females to basically induce a female to come and mate with them. And so they are basically putting out sound 24 hours a day practically, and it is a very energy intensive sort of activity. So not surprising that you would have the muscles associated with producing vocalization to be oxidative. While they're doing that, they're basically sitting around in a pond, not really moving all that much. And so one of the things that you'll see is that the skeletal muscles of amphibians and reptiles are dominated once again by these glycolytic fibers. These two different types of fibers are called oxidative and glycolytic because of the type of ATP production that goes on in them. So when we think about metabolic rate, we can think about resting metabolism, which is the metabolism that you possess when you are not digesting food and you're not doing anything. And so for you, your resting metabolism would be measured as you're laying in bed. First thing in the morning, you are awake, but you have not gotten up and you haven't had anything to eat since the night before. And then exercise metabolism is the metabolism that you experience when you are actively exercising. And that can vary a lot depending on what the level of metabolism is. And then there's this idea that they separate in your textbook about field metabolism, which is kind of the average metabolism that an organism would experience in their daily activities. And this would both be a combination of both exercise metabolism and resting metabolism. So field metabolism would be kind of a longer term average metabolism for an organism. Normally, when a physiologist is trying to measure metabolism, if they want to know, like, does one species have a higher metabolism than another, you would be measuring resting metabolism, even though that is not necessarily the metabolism that is most important for example, the fitness of an individual. So glycolytic metabolism makes up about 80% of ATP production in herps. And once again, the problem with this is that it produces lactic acid as a byproduct, and this can build up relatively quickly. And so what happens as you increase the speed of an organism? So if you are a lizard out in a landscape and you're basically out there foraging, and if you're like a teid lizard, the way a teid lizard forages is it's looking around for insects. It's flipping over small stones, looking under them, and then it experiences these short darts of sprinting from one place to another place, where it then stops and once again digs around and looks for food. And so it's these little short, tiny bursts of energy followed by kind of lower level activity, such that its speed through the environment is not particularly fast. Well, a teid lizard can keep up those slow speeds for long periods of time because it's down in this oxidative metabolism area. Its speed is not so fast that it's requiring more energy than it can liberate through oxidative metabolism. Remember that oxidative metabolism is efficient, but it's also a little bit slower a method to create ATP. If, on the other hand, that lizard starts to be chased by another lizard or starts to be chased by a hawk or something like that, it will ramp up its speed very quickly. And as it ramps up its speed very quickly, it goes beyond what aerobic metabolism can handle and enters anaerobic metabolism. At that point, it's using up more energy than oxidative metabolism can offer, so it enters glycolytic metabolism to augment oxidative metabolism. And so total metabolic output is higher because you're operating at these higher speeds, but you have gone past your maximum aerobic speed. And as a result, you're now creating more energy, but the extra energy that you're creating is less efficient, and it's also causing this buildup of lactic acid. And that ultimately becomes non-sustainable. You can't continue to sustain that over long periods of time. So I bike a lot. And if you bike a lot, and if you're planning on biking long distances, one of the things that you can go to your exercise physiologist and get measured is your VO2 max. Do you guys know what the VO2 max is? Anybody? You guys don't go to your exercise physiologist for this? It's basically the maximum speed that you can reach while maintaining aerobic metabolism. And so essentially, how much aerobic capacity do you have? And so if you talk to professional cyclists in particular, one of the things that their physiologists are paying attention to is what is a particular cyclist VO2 max. The higher that is, the longer they can stay in aerobic metabolism before verging over into anaerobic metabolism. And you can alter this quite a lot simply through good training. So for example, I'm planning on doing a thing called the triple bypass in August. You guys are probably not familiar with triple bypass. Triple bypass, it's a bike ride in Colorado. It starts in Evergreen, Evergreen, Colorado, and goes to Aurora, Colorado. And it goes over Squaw Pass, and then Loveland Pass, and then Vail Pass. So it's called the triple bypass because you go over 3 mountain passes. It's 117.5 miles, and you climb 10,000 feet of climbing in the process of doing this. I did it 2 years ago. I was scheduled to do it last year, again, with a friend of mine who didn't finish it 2 years ago, but it was canceled. So now we're going this year. And so one of the things that I'm working at is to basically take this red line for me and move this red line to the right, so that as I have to exert more and more energy to make it over these mountain passes, I minimize the time that I am exercising in this anaerobic area up here, so that I am basically staying in aerobic oxidative metabolism as long as I can on this ride. Because as soon as I start entering glycolytic metabolism, I know that I'm approaching the end of my physiological ability to do whatever it is that I'm in the process of doing. And you can increase your maximum aerobic speed by simply training in ways that concentrate on doing that. And of course, this is especially problematic because in Colorado, we're basically bicycling at high elevations. So Loveland Pass, which is the second pass that you go over, is at about 12,000 feet elevation. So there's less oxygen up there. And so if I'm not training in a way to increase my VO2 max, then I'm going to suffer badly when I get to Colorado in August. So I'm training right now in ways that will move this line to the right. Normally, we don't think of an amphibian or a reptile as in training for anything, but we can actually measure what the maximum aerobic speed is of individuals and look and see how that varies across species. And what we find is that for some species, their VO2 max is higher than other species. And those individuals are individuals who have the capacity for longer periods of aerobic activity. And they can sustain those longer because they can achieve higher speeds, more activity at levels that are still within the aerobic range of activity before they have to enter glycolytic metabolism. Amphibians and reptiles in general, though, are known for having relatively low levels of maximum aerobic speed. They can get to fast speeds, but they get that by sacrificing efficiency because they sustain that speed using glycolytic metabolism. So as they're doing this, they can convert a lot of that lactic acid back into glycogen, which allows them to recover quickly. And so what you generally see in amphibians and reptiles is you see brief periods of intense activity followed by brief periods of rest, followed by brief periods of intense activity, followed by brief periods of rest. And so if you watch a lizard moving through a landscape, this is what you're going to see. Full out running, followed by stopping and resting for a few minutes, followed by full out running, followed by stopping and resting. Because they're achieving these maximum speeds, because they undergo glycolytic metabolism. But one of the nice things is they're very good at recovering from those. So they have potentially short bursts of activity, but also relatively quick recovery times. So a story to illustrate this is when I was in graduate school, we went on a field trip to South Texas. And in South Texas, there is a species of lizard that is pretty rare. It's the reticulated collared lizard. Let me find a picture of one. It's a very pretty lizard. It's got this tan and black coloration with a lot of yellow on it. They're fairly large lizards, but they only occur in this one little section of extreme Southwest Texas. And when we took this field trip, one of my goals was to catch this species and take photos of it. And so me and my friend Dan Sines were out collecting on this field trip. And Dan flipped a rock, and there was a colored lizard under it. It was a reticulate colored lizard, and he grabbed for it and missed it. And that guy just hauled ass. It got up, it started running bipedally, and it was just off. And I took off after it because he was there standing holding a rock. And it was way faster than I was. But one of the things that I knew about this is if I just kept the pressure up on this lizard, it was going to eventually tire out. And even though it was faster than me in the short sprint, because it very quickly left aerobic oxidation, oxidative metabolism rather, and was very quickly driving itself into glycolytic metabolism, I knew that because I was going to spend my entire energetic effort on chasing that lizard down in oxidative metabolism, I knew that I had way more endurance than he did. And so I was chasing him across this desert flat. He ran probably 40 yards. And then I'm sure he felt himself coming to his physiological limits, and he dived underneath a rock. And I caught up to him. I saw where he went underneath the rock. I flipped the rock. Guess what he didn't do?
Run.
He didn't haul ass running because he had exhausted all of his energy supplies through that short burst of energy. And I got to the rock before he could recover any of that. And so I flipped the rock, grabbed the lizard, put him in the bag, took him back to camp and took photographs of him, and then released him because I knew what his physiology was like. I knew he was going to have this short burst of energy, and then I knew that he would have to have a refractory period to recover that energy. And if I could get to that rock during that period, I would be much more able to collect him than if he had greater aerobic capacity and could have sustained that speed for a longer period of time. And I just flipped over the rock, looked at him, reached down, grabbed him, and stuffed him in a pillowcase. So this is the reason that your book goes into this distinction between these two types of metabolism is it has profound effects for the ability of these animals to avoid predation. As long as all you have to do to avoid predation is sprint from one rock to another rock, that's all you really need. Because once you're underneath that other rock, you're safe unless you're going up against a herpetologist who can just pick the rock up and nab you. And so there are good reasons why why herps lack the aerobic capacity that other organisms do. And yes, Kelsey, you're very welcome. Happy to use the term herp for you.
Still don't like it.
All right. On Friday, we will finish up the last little bit on metabolism. We don't really have that much more to go. And we will talk about the Buckley and Huey paper after we're done with covering metabolism. Questions? All right, then I will see you guys tomorrow in lab to continue working on reptile identification.